So here is the third and final chunk of the nitrogen fixation and amino acid metabolism lecture series, the last bit here of chapter 23. We've already talked about nitrogen in general. We talked briefly about the nitrogen cycle and how we get nitrogen out of the atmosphere as molecular nitrogen into gas and into living systems. And in the last chunk, we talked quite a bit about the anabolism of nitrogen-containing compounds, focusing almost exclusively on the amino acids. So we learned how amino acids are built, kind of as a superficial overview. We ended that discussion talking about the amino acids we can't build. These are the essential amino acids and where we get them from and how they play important roles in our bodies. So this last lecture chunk is all about the other side of the metabolism of amino acids, the catabolism, or the breaking down of amino acids, and that's where we'll wrap up this story right now. So breaking down amino acids again is catabolism, and the first real step that we need to be concerned about when we break amino acids down is what got us into this mess in the first place. We got to do a transamination reaction because we got to make sure that that amino group, essentially that ammonia molecule, that ammonia ion, is taken care of. So monitoring and regulating the ammonia is the critical part of all of this for the cell. So that amino group is offloaded off of the amino acid as it's being broken down or digested, and it is put on our amino group shuttle, alpha-ketoglutarate. Once alpha-ketoglutarate accepts that ammonia molecule, we've made glutamate, the amino acid glutamate. And we talked about the alpha-ketoglutarate story extensively in the last lecture chunk. So it's the same thing. So alpha-ketoglutarate is the universal acceptor of nitrogen in our bodies. That's a transamination reaction, and that's how we always shuttle ammonia around in our cells. What we're left with after that amino group has been donated to alpha-ketoglutarate is what we call the carbon skeleton of the amino acid. Really, it's just the carbons and the groups linked onto those carbons. That's all that's left. So we're going to kind of take a fork in the road now. The amino group goes one way, and the carbon skeleton goes the other way, and we're going to focus on the carbon skeleton first. But remember, as we go through this piecemeal, we just took an amino acid and basically split it in half. The amino group went one way, and the rest of the molecule went the other. So don't forget that these two components came from the same single amino acid that we are digesting and catabolizing here. So carbon skeletons of amino acids can go down one of two metabolic roads, and we call those glucogenic and ketogenic. A glucogenic amino acid has a carbon skeleton that can be used to make either pyruvate or oxaloacetate. Remember when we talked about gluconeogenesis, oxaloacetate was the very first precursor for going backwards in glycolysis. Pyruvate became oxaloacetate, oxaloacetate became PEP, and then down the road we went all the way to glucose. So these amino acid skeletons, these carbon skeletons of these amino acids, can eventually be made into glucose. That's why we call them glucogenic, because they can be fed into gluconeogenesis. Ketogenic carbon skeletons of amino acids can become acetyl-CoA, or another related molecule which we don't really discuss in this class, acetyl-CoA. That's involved in lipid metabolism. But it can go down into the citric acid cycle, essentially. Sooner or later, these carbon skeletons will be used to make ketone bodies. That's where we get the name ketogenic. But again, from our purposes, they will be fed into the citric acid cycle so that electrons can be stolen from them. Amino acid carbon skeletons can also be siphoned off of these metabolic pathways and be used as precursors or intermediates for making other molecules that we've encountered before, such as alpha-ketoglutarate, fumarate, succinyl-CoA, heme, lots of the things that we've already talked about. So I've shown this diagram a few times already, but it's important to show it again. The citric acid cycle truly is the hub of all of metabolism. In the last lecture chunk, we talked about alpha-ketoglutarate being made into glutamate. Glutamate can accept a second amino group to become glutamine. We can make other molecules from there. We also talked a little bit about oxaloacetate becoming aspartate and asparagine. And we talked quite a bit about the serine family, how we can make serine and cysteine and glycine as well. So we can make amino acids from the citric acid cycle. But here what we're talking about is feeding amino acids into the citric acid cycle. So the carbon skeletons of glutamine or glutamate can be made into alpha-ketoglutarate. The carbon skeletons of tyrosine and phenylalanine can be used to make fumarate. And so we can feed molecules into the citric acid cycle as well. And most amino acids can, one way or another, become acetyl-CoA if they're ketogenic, and that acetyl-CoA doesn't become part of the citric acid cycle. It's fed into the citric acid cycle as a raw material so that electrons can be stolen from it, electrons that can eventually be used to pump protons and, of course, be made into ATP. So essentially, speaking metabolically, the glucogenic amino acids can be used to make glucose, which can then be converted into glycogen for storage. And the ketogenic amino acids can be used to make acetyl-CoA, which is fed into the citric acid cycle and eventually used to generate ATP. One way or another, we can harvest energy from these molecules. It's whether or not that energy can be stored that's different. In general, we have a lot more glucogenic amino acids than we have ketogenic. Only leucine and lysine are exclusively ketogenic. Some amino acids can go either way, such as isoleucine, phenylalanine, and the others that are listed. But you can see that most amino acids, the vast majority of amino acids, are glucogenic and can be fed into gluconeogenesis. So that's what happens to the carbon skeleton portion of amino acids. What about that ammonia? What about that amino group that we offloaded to alpha-ketoglutarate? What happens to that? Well, excess nitrogen in the form of ammonia that's no longer needed by the body can be excreted and is excreted as metabolic waste. As far as living systems on this planet go, that excretion can happen in the form of ammonia, urea, or uric acid. And some animals can use a combination of those different forms. Aquatic animals, most notably fish, they actually do excrete straight ammonia, and they can get away with it because they're living in large watery environments. The ammonia that they excrete through their skin is rapidly carried away and diffused into that watery environment, and they're not toxifying themselves because that ammonia gets carried away with the water. Most terrestrial animals, including ourselves, we excrete nitrogen primarily as urea. That's our predominant way of getting rid of ammonia waste. Urea is less toxic than ammonia. It is water-soluble, so it'll dissolve in water, and it can be passed as a dissolved-in-water molecule. But that means making pee. And so if we're going to be dissolving all of our urea in water to make urine, that means we need to carry a lot of water around with us in order to have that urea have a place to be dissolved in. So not every animal on this planet can get away with carrying surpluses of water around. Can you think of an animal that can't afford to be carrying around the weight of all this excess water that would be needed to dissolve all this urea? Birds. Birds need to be as light as possible. In fact, if you don't know it yet, the bones of birds are actually hollow. Birds have hollow bones. So birds have evolved to be as light as possible, making flight as easy as possible. And birds can't be carrying around all this extra unneeded water to dissolve their urea in. So birds excrete their nitrogen waste as uric acid, a solid that needs very, very little water to be passed. In fact, when you think of bird poop on your car, the white portion, the majority portion of bird poop is actually bird urine, not poop at all. It is the uric acid that requires very little water to allow those birds to be as light as possible. Incidentally, that's why the danger exists for a very nice car paint job when there's too much bird poop on it. That uric acid actually eats away at the paint of the car. It could leave a permanent stain behind. So that's an introduction into nitrogen waste, but let's get into the biochemistry now. The urea cycle is the last true cycle that we'll talk about in non-photosynthetic organisms, and the urea cycle exists solely as a urea generator. This cycle is used to gather all excess cellular nitrogen and use that nitrogen to make urea, this less toxic and water-soluble form of ammonia waste. And then eventually that dissolved urea will be excreted by the organism or the animal as urine. So the nitrogen that enters this cycle comes from our universal ammonia shuttle, glutamate. Glutamate comes to the urea cycle. It offloads an ammonia molecule. It offloads an amino group. That allows that glutamate to revert back into being alpha-ketoglutarate. And that reaction is catalyzed by our very old friend, glutamate dehydrogenase, the same enzyme that made glutamate for us in the first place. Nothing new here. But this glutamate, in this situation, actually received that ammonia molecule, it received that amino group from who knows where in the body. picked it up as an excess ammonia that was no longer needed, picked it up as that excess ammonia that was released as an amino acid was being catabolized, and it brought that ammonia molecule here to the urea cycle where it could be offloaded. Part of this urea cycle occurs in the mitochondria, the rest is cytoplasmic, the beginning stages are mitochondrial. So there is an enzyme called mitochondrial glutaminase, which can also extract one of the two amino groups from a glutamine and provide free ammonia to the urea cycle that way as well. This is a minor delivery mechanism to the urea cycle, but we can get ammonia to the urea cycle through glutamine as well. So let's do it. Let's make some P. So the very first reaction of the urea cycle is a condensation reaction. We're familiar with condensation reactions not only from the citric acid cycle, but I'm sure from your previous coursework as well. And that's a condensation reaction that occurs between ammonia, the ammonia that was just offloaded by glutamate, and carbon dioxide. So when you condense ammonia with carbon dioxide, you make a molecule called carbamoyl phosphate. Yes, we pick up some phosphates as well, and those phosphates come from ATP. The energy for this reaction also comes from ATP. We are overly organizing these molecules, we're reducing entropy, and so energy is required to make these reactions occur. The cell readily gives up its ATP for this process, however, because accumulating ammonia would be toxic for the cell, and so getting rid of nitrogen is a priority. It's worth burning the ATP to get rid of this nitrogen waste. Carbamoyl phosphate is the entry point into the urea cycle. You can think of carbamoyl phosphate as analogous to acetyl-CoA. The way that acetyl entered the citric acid cycle, carbamoyl phosphate enters the urea cycle. And just as acetyl complexed with oxaloacetate in step 1 of the citric acid cycle, carbamoyl phosphate is going to complex with ornithine in step 1 of the urea cycle. So carbamoyl phosphate, which is a delivery mechanism for urea waste, for amino acid waste, for ammonia waste, reacts with ornithine, and that forms a molecule called citrulline. Once we make citrulline, we leave the mitochondria and the rest of the urea cycle becomes cytoplasmic. So we're right here in the early phases of the urea cycle. Here is ornithine, here is carbamoyl phosphate, there's the amino group it's carrying, that's the ammonia that it is carrying, and carbamoyl phosphate and ornithine come together to form a molecule called citrulline. Again, the first step of the urea cycle. That citrulline leaves the mitochondria, becomes cytoplasmic, and then aspartate, an entire aspartate amino acid, reacts with and joins up with citrulline, and this forms a beast of a molecule called arginosuccinate. Arginosuccinate is large, very, very large, and you'll see that it is carrying multiple nitrogen-containing groups. 1, 2, 3, 4 nitrogen-containing groups here. So we've brought at least another full ammonia to the party, as well as the nitrogen groups that were initially carried by aspartate. We also burn ATP to drive this reaction forward. Arginosuccinate, this very large beast, is then split. It's cleaved to form arginine, and this is the arginine that I referred to in the last lecture chunk where I said technically arginine is not an essential amino acid, but we need to consume a lot of it because most of the arginine we do have is dedicated to the urea cycle. So this is that arginine that's really exclusively dedicated to the urea cycle. But we also make fumarate in this step as well. When that arginosuccinate is cleaved, it's cleaved into arginine and fumarate. That fumarate is a leaving group. It actually leaves the urea cycle as a byproduct of that reaction. So it's arginine that's going to continue on in the urea cycle. Finally, that arginine is hydrolyzed. When it's hydrolyzed, urea is yielded. That's what we were after all along. And the other half of hydrolyzed arginine is ornithine. Ornithine is the entry point, or the central intermediate of the urea cycle. And we've returned back to the beginning. That ornithine will be transported back into the mitochondria, where it is ready to complex with another carbamoyl phosphate. Remember that carbamoyl phosphate was made by accepting ammonia, carbon dioxide, and ATP, energy from ATP. That ornithine, once complexed with carbamoyl phosphate, will become citrulline. Citrulline will leave the mitochondria and join up with an aspartate to give us arginosuccinate. Arginosuccinate will be cleaved into fumarate, which leaves, and arginine, which continues. That arginine will be hydrated and cleaved through hydrolysis to yield urea and ornithine. So it's a very simple cycle compared to the citric acid cycle, really just a total of 4 steps. to regenerate ornithine. Step 1, the complex with carbamoyl phosphate. Step 2, the production of arginosuccinate when aspartate joins. Step 3, the cleavage of arginosuccinate to fumarate and arginine. And step 4, the cleavage of arginine to ornithine and urea. Fumarate is shared between the urea cycle and the citric acid cycle, so we can think of fumarate as a link or a form of communication between these two cycles. As one cycle increases its speed and the amount of fumarate grows, because of that cycle being that much more active, it can communicate that higher rate of activity with a neighboring cycle of another kind of linking those two together. So if one cycle, say the citric acid cycle, is going strong and has plenty of fumarate as a result, and now we build up nitrogenous waste, we build up ammonia waste, some of that fumarate can kind of be donated over to the urea cycle and feed its intermediates to allow that other cycle to speed up. What we often see metabolically after a high protein meal, of course, if we've eaten a lot of protein in our diet, we have a lot of nitrogen waste, a lot of ammonia waste because of all those amino acids in that high protein meal. That cell, that body will likely have too much nitrogen, and so the citric acid cycle will speed up because the urea cycle is so active that it donates a lot of that fumarate to new citric acid cycles, getting rid of the excess nitrogen and also allowing the body to metabolize those carbon skeletons and get some energy out of it as well. In humans and other higher eukaryotes, the urea cycle is restricted to the liver, but all the same, it is this cellular mechanism to get rid of ammonia and detoxify the environment that is the most interesting in my opinion. So I've shown this once before, but this is the metabolic map of a eukaryotic cell. This central corridor is glucose metabolism, starting with glucose, going through glycolysis. The PDC is represented here, and here is the citric acid cycle with the electron transport chain and ATP production here at the end. And if we look over here as an offshoot of this central hub, we see a second cycle, primary cycle, and that is the urea cycle. Here we have ornithine to citrulline, citrulline to arginosuccinate, arginosuccinate to arginine and fumarate. Fumarate can crosstalk between the two here, and then back to ornithine and urea, which is going to be secreted as urine. That urea dissolved in excess water by the kidneys and then secreted through the bladder as urine. Ornithine ready to get another carbamoyl phosphate and repeat the cycle as well. And we also see further linkages here because we can make aspartate from oxaloacetate, and we need aspartate for the urea cycle. So these cycles are not only linked through fumarate, they're linked by aspartate as well. All of these processes are, of course, linked. The metabolic map shows that there's tons of crosstalk between all of these mechanisms. So to summarize this last chunk, the first real step of catabolizing any amino acid is taking care of that ammonia. Get that amino group off as ammonia and quickly give it to alpha-ketoglutarate so that it is safe and that glutamate can shuttle that amino group anywhere it needs to go. Once you have removed the amino group from the amino acid, you're left with a carbon skeleton, and that carbon skeleton can go down one of two primary roads. It can be glucogenic, which means it can be made into pyruvate, oxaloacetate, and eventually into glucose by the gluconeogenesis process, or it can be ketogenic, where the carbon skeleton is used to make acetyl-CoA, which can be fed into the citric acid cycle to yield electrons and eventually ATP. Either way, those carbon skeletons can be used for energy, either directly as acetyl-CoA or indirectly via glucose. Back to that amino group, though, that we offloaded, the urea cycle is used to gather all of those excess amino groups and use them to create urea, a molecule that can be dissolved in water and excreted as urine. It's that urea cycle that we spent a decent amount of time on here, going from ammonia to carbamoyl phosphate, complexed with ornithine to create citrulline. Citrulline joins up with aspartate to give you arginosuccinate. That molecule cleaved to yield arginine and fumarate, and arginine hydrolyzed back to ornithine and the urea we were after all along. So that wraps up all of nitrogen metabolism, or at least insofar as we're going to cover it in this course. That's also the end of the Lecture 18 series, leaving us only photosynthesis to cover now for the rest of this course, and photosynthesis will be broken up over two lectures. Lecture 19 will be the light reactions, and Lecture 20, the final lecture of the course, will be the dark reactions.